I'd like to greet everyone. Good afternoon, and welcome to our webinar. We are very glad to have you here today. The webinar is dedicated to discussing an important topic entitled Beyond Organics, the Science of Biostimulants and Biopesticides. My name is Martina, and I'll be your host for today. First, I would like to extend our warm thank you to our webinar chairs, Professor Dr. Youssef Rufal and Professor Dr. Giandomenico Corrado, and our speakers, Dr. Michele Ciriello, Professor Dr. Petronia Carillo, and Professor Dr. Luca Ruio. Before we begin with the presentations, I would like to briefly introduce NDPI, the publisher behind this event. NDPI is a pioneering scholarly open access publishing. Since 1996, we have been committed to supporting academic communities worldwide. Headquartered in Basel, Switzerland, our mission is to foster open scientific exchange in all forms and across all disciplines. Today, NDPI publishes more than 490 open access journals. The organizer of today's webinar is the journal Agronomy, international peer-reviewed open access journal on agronomy and agroecology. The journal is published monthly by NDPI. We will have 3 presentations, each approximately 20 minutes long, followed by a dedicated questions and answers session and a brief closing of the webinar. If you're interested in topical collection related to today's topic, you'll find a hyperlink within the presentation. And I will also post the link in the chat. There you can read more about our current call for papers. Before I hand the floor over to our guests, I would like to encourage everyone to submit your questions through the questions and answers box at the bottom of your screen. If you prefer to type them in the chat, please make sure you select everyone so that we can see them and transfer them into the questions and answers section. When asking your question, kindly indicate which speakers it is addressed to. I would also like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible. And now a very warm welcome to our chairs, Professor Dr. Youssef Rufal from the Department of Agricultural Sciences, University of Naples, and Professor Dr. Giandomenico Corrado from the Department of Agricultural Sciences, University of Naples, Federico II. Also, I am delighted to welcome our speakers, Dr. Michele Ciriello from the Department of Agricultural Sciences, University of Naples, Federico II, Professor Dr. Petronia Carillo from the Department of Environmental Sciences and Technologies, University of Campania, Luigi Van Vitelli, and Professor Dr. Luca Ruio from the Department of Agricultural Sciences, University of Sassari. Thank you very much for being part of this event. We are delighted to have you with us. That is all from my side, and now I hand over the microphone to our chairs, and I hope you all enjoy our webinar. Professor Youssef Rufal, feel free to start the introduction when you're ready.
Hey, thank you, Martina, for this nice presentation. Good morning or good afternoon to everyone. And welcome to our webinar in agronomy concerning organic horticulture and agriculture, with a particular focus on plant biostimulants and biopesticides. This webinar supports, as the participants know, the European Green Deal goal for organic farming by 2030, promoting sustainable agriculture through an innovative hybrid solution, not only the use of biostimulants and biopesticide but combining biostimulants with microbial biopesticides to enhance plant resilience, to manage pests, and to boost yields and sustainability. I would like to say that this event, this webinar, is a part of our national project funded by the Italian Ministry of Education, University and Research, with entitled Exploring Synergies Between Biostimulants and Pesticides for Increasing Productivity and Sustainability in Organic Horticulture. During this webinar, Professor Gian Domenico Corrado from University of Naples, Federico II, will share the whole session and will introduce our three speakers. Thank you to all the participants for joining us, and let's begin. Please, Professor Corrado, the floor is yours.
Okay, thank you. Thank you, Joe, for your clear overview of the context. I think we have a tight schedule, but as Martina said, before we begin our, just a quick reminder to our audience. So this is an interactive session, so feel free at any time to type your questions in the Q&A box, but then Martina will collect them and then we will address them at the end of the session. So please stay tuned until the end of the sessions, especially if you are interested in your questions and the answers. So let's move to our first presentation, and I'm very pleased to introduce Dr. Michele Ciriello. He's a postdoctoral researcher at the Department of Agricultural Sciences at the University of Naples, Federico II, and his work focuses on understanding the morphological, physiological mechanisms that are activated by biostimulants, especially non-microbial biostimulants, and how biostimulants can help plants to cope with adverse abiotic conditions. So that's enough, and Michele, the floor is yours.
Hello everyone, I'm Michele Ciriello, a postdoc researcher at University of Naples Federico II, and as suggested by the title of my presentation, the aim of my speech is to investigate how the interactive effect between the application of bioproducts such as biostimulants and biopesticides can influence the productivity of vegetable production. Currently, the agricultural sector is being colored up to produce more for a constantly growing population. However, the challenges imposed by climate change and increased biotic pressure are accelerating the need for a profound change in the entire agri-industrial sector. The Sustainable Development Goal defined by the United Nations, in addition to following the goal of eliminating world hunger, emphasize the need to do it so while respecting the vitality and the quality of the resource of our planet. And in this specific context, one of the most important challenges is to try to reduce the use of synthetic agrochemicals, such as fertilizer and pesticides, due to their documented negative impact on environmental and human health. The European Green Deal has set a target of allocating 25% of agricultural land to organic farming by 2030, as said before by Professor Ruffel, and according to the definition, organic farming is an holistic production management method that maintains and enhances ecosystem health, including biodiversity, biological cycles, and biological soil activity. But in any case, organic farming is mainly characterized by a chronic lack of production capacity. This is mainly related to the impossibility of using synthetic agrochemicals and at the same time the inability of standardized bioproduct on market. Despite this, the graph presented in this slide clearly show how in the recent year, more and more agricultural land has been allocated to organic farming. In fact, we can see a constant increase over time. Still talking about numbers, we can see that the country with the largest area of land allocated to organic farming is represented by Australia, followed by India and Argentina. It's interesting to know that Italy, despite its modest size, allocates more agricultural land to organic farming than the United States. Even though the numbers show growth and constant interest in organic production driven by the growing consumer interest in this field, the problems of production in organic context undermine the sustainability of this type of agriculture. As we discussed before, the impossibility of using synthetic agrochemicals drastically reduce the potential yield. And taking into account the data in the literature, we can say that on average, organic production has a production gap of about 25% compared to the conventional cultivation method. At this point, it's necessary to find strategies and tools that can reduce this gap in order to make organic production more sustainable. In an organic farming context, the application of biostimulants products could represent a very interesting alternative to synthetic fertilizer. And in recent years, scientific research has shown that the application of these bioproducts can increase resource use efficiency, mitigate at the same time the negative effect of abiotic stress, and improve the quality of agricultural products. In accordance with the European regulation, a plant biostimulants means as European fertilizing product stimulating plant nutritional process independently of the product's nutrient content, with the sole aim of improving one or more of the following characteristic of plant and the plant rhizosphere as well. These are nutrient use efficiency, the tolerance to abiotic stress, crop quality traits, or availability of confined nutrient in the soil or rhizosphere. It's important to emphasize that the ultimate function of biostimulant is not to directly supply nutrients to plant or protect their production from biotic stressor. In fact, the biostimulants are not fertilizer, but a new tool that can be used to improve agricultural production. After several times explaining what biostimulants are not, taking the European regulation as a reference, this slide shows the many classification of biostimulants products, and in particular, biostimulants are classified according to their origin into microbial and non-microbial products. Microbial products include mainly PGPR and beneficial fungi, while for the non-microbial products often refer to as substances include the protein hydrolysates from animal or vegetable derived, humic acid and seaweed and plant extract. And regardless of the origin and the classification of the biostimulant, the importance of this bioproduct is not limited to the world of research. In fact, the data on the global biostimulant market predict future growth in the coming years, demonstrating the economic interest in this product. A more in-depth analysis showed that humic acid and protein hydrolysase are the most popular types on the market, and Europe is the undisputed leader in the use of biostimulant. However, when we look at the most common method of application for the biostimulants, we see that these products are mainly used by foliar application. Even though the economic data, it's important to know that the positive response to biostimulants also depend on how they interact with the genotype and the growing environment. In addition, the effectiveness and the positive response to biostimulants depend on the other fundamental critical aspect that must be optimized. In particular, the type of biostimulant, the dose used, the formulation, the frequency of application, and obviously the method of application. This is confirmed by data collected in a recent meta-analysis. Specifically, we see how the choice of biostimulants influences production response. Among the various target crop, we see that the application of biostimulant has a more positive influence on vegetables. Growing conditions have a strong impact on the action of biostimulant. In fact, we see that in percentage terms, yield increase are higher in arid environment and in a condition of moderate and or high salinity. In any case, the data collected by the authors summarize how yield increase strongly vary based on different aspects. Even though biostimulant can lead to significant increase in yield, it's important to mention the main downside that need to be solved in the near future. Regardless of the origin, biostimulants are really variable. This variability, which often leads to no standard results, is due to the different origin of the starting materials, which are often waste materials or different type of organic matter, and of course also to the production process. As mentioned above, another distinctive feature of biostimulants is that their positive effects are strongly influenced by the interaction of various pre-harvest factors that are not always controllable. These include, for example, the target crop, the phenological stage, and soil and climate condition. And in addition to these, the biostimulant action is attributable to the co-presence of bioactive molecules or consortia of microbes, which make it difficult to identify the actual and punctual mechanism of action. Moving our attention to another category of bioproducts that are essential in organic production context, we begin to talk about biopesticides. Biopesticides are a sustainable complement and or alternative to synthetic pesticides, and these products are derived from natural substances and biochemicals, which in nature have key ecological activities in controlling pests. As with fertilizer, current policy call for a reduction in the use of synthetic pesticides. Many of the chemical pesticides used in agriculture are synthesized from hazardous chemicals. These chemicals have adverse effect on human health and cause environmental pollution. They are also harmful to no-target organisms. They contaminate water and soil and they can cause bioaccumulation. In response to these, the biopesticides are synthesized from organic compounds and are therefore not harmful to the environment. Specifically, they are not responsible for pollution resources such as water and not target living organisms. However, as with biostimulant, we cannot ignore the current disadvantage of these biopesticides. The production process, similarly to the biostimulants for biopesticides, is very complex. The production of biopesticides requires significant investment and is also time-consuming. In addition to this, biopesticides require selection, development, and authorization by regulatory authorities for the production of the new biological agents. Farmers must use multiple biological agents to control different pathogens and pests in the field, and the positive action of biopesticides can be compromised by adverse environmental conditions. The end, so biopesticides have a short shelf life due to the sensitivity of fluctuation in temperature and humidity. So biopesticides as we classify according to their sources of extraction and the molecules or compounds used in their preparation. Microbial pesticides are obtained from microorganisms such as fungi, bacteria, or virus. The active molecules or compounds isolated from these organisms attack specific species of pests of entomopathogenic nematodes. The most commonly used biopesticides are living organisms. These include biofungicides, bioherbicides, and the bioinsecticides. Then we have biochemical pesticides, which are natural products used to control pests through non-toxic mechanism. They are classified into different types such as pheromones, plant extract, or oil, and natural insect growth regulation. As with biostimulant, the biopesticides market is also growing economically, reflecting an ever-increasing interest in the use of bioproducts for the control of biotic agents in agriculture. So considering these, future innovation in sustainable agriculture could involve the use of bioproducts, but in a hybrid formulation. This challenge was at the heart of our research project called BBORT, and specifically, we hypothesized that specific hybrid biopreparation by stimulant and biopesticides could provide synergistic benefit to their compatibility and independence. To validate our hypothesis, we conduct an experiment on organic tomatoes treated with single and combined application of biostimulant and biopesticides. Taking into account literature, the possible synergy between the two byproducts could be linked to the combined action of the mechanism of action of the two bioproducts, which will trigger higher reactivity to pests thanks to the activation of different response. Specifically, biostimulants can improve the growth and persistence of biopesticides, thereby increasing their effectiveness and improving the efficiency of nutrient use in treated plant. The research project in question lasted 2 years, and in the first years we evaluate the effect of the byproducts individually or in combination on the vegetative growth and physiological response of tomato plants. In the second year, we investigated the productive and yield response following the individual or combined application of the two byproducts under study. As I said, the experiment was conducted on tomato grown in greenhouse using organic farming methods. The treatment studied were an untreated control, further application of plant-based protein hydrolysate, further application of an entomopathogenic fungus, and a combination of the two. Both byproducts were used at the recommended dose, and the different treatments were replicated 3 times. Each replication consists of 10 plants. Turning to the results, we can see that the data collected in the first years confirm that the application of the protein hydrolysate biostimulants improved the vegetative growth of the treated plant. Specifically, single or combined application increased the dry biomass on the plant by almost 50% compared to the control plant. Even though the biopesticides didn't make a big differences in the biomass production, these graphs show how all the treatment improved the physiological response, specifically the net assimilation of carbon dioxide. Gas exchange measurements were taken on healthy leaves and repeated several times during the experiment. The data reported here refer to the measurements taken 30 days after transplanting. At the end of the first year of the testing, leaf samples were collected to determine their ionic content. The graphs represented in this slide show the results for nitrate, phosphate, and potassium content. The greatest differences can be seen in nitrate, where the application of the biostimulant significantly increased the yield, the leaf content. These results highlight the positive role of the biostimulant in organic farming context where key elements such as nitrogen can be limiting. These results will in fact justify the better vegetative performance obtained by plants treated with the biostimulants applied individually or in combination with the biopesticide. With regard to potassium and phosphate content, no significant differences were observed compared to the control. Nevertheless, numerically, we can observe an increase for both parameters for plants subject to the combined application of bioproducts. The positive effect of vegetative growth and photosynthetic performance of plant treated with the biostimulant could justify the better production results obtained in the second year by plant treated with a single or combined application of the biostimulant. Specifically, we observed a 50% improvement compared to the control plant following the application of the biostimulant under test. So the application of the biopesticide did not result in a significant differences compared to the control. So the improvement in yield observed in plant treated with a biostimulant was not attributable to a greater number of fruit per plants. In fact, no significant differences were observed between treatments for these important biometric parameters. The reason for the better yield was in fact linked to the higher fresh weight per units of the fruits of plant treated with a biostimulant. So in conclusion, we can say that the combination of biopesticide and biostimulant has had modest or not significant effect on morphophysiological traits and mineral composition compared to their individual application. However, this combined approach did not perform worse than the sum of their effect, indicating a positive outcome. Bio-based bioproducts notably improve the yield and mineral composition of greenhouse grow organic tomatoes. The foliar application of biostimulants allow or combined with the biopesticides enhance photosynthesis and mineral status, leading to increased crop productivity. Additionally, use these bioproducts together can improve labor efficiency, making it a practical option for organic farming. So with this, thank you so much.
Okay, thank you, Michele, for the overview and for the very rich presentation. Thank you. And moving on with our program, now it's a pleasure to welcome our second keynote speaker, Professor Petronia Carillo. Sorry. She's a full professor of agronomy at University of Campania Luigi Vanvitelli, where she teaches plant physiology and agronomy, and her scientific approach studies combines metabolomic profiling with physiological studies, and she works especially on understand how plant physiology adapts to abiotic stress and to biostimulants. So we are very keen to hear what she has to say about physiological mechanisms behind stress tolerance and benefits of biostimulants. So Petronia, please go ahead.
Thank you for your presentation. I decided not to share our data. I have provided metabolomic data that profile of metabolites to both groups, the other groups with which I am collaborating for this project, but I want to speak about the effect of plant stress on plants, in particular of two main stress that are drought and salinity, and what the biostimulants can do against these stresses, helping plants to cope with them. In the last period, climate change is accelerating and amplifying the effect of these two stresses, because the increase and the rise of average temperature is declining summer and also winter rainfall, causing a build-up of drought and salinity. From a first study of 1918, it seems that drought causes 83% of all climate-related agricultural losses, and soil salinization, that is also a consequence, in part a consequence of drought, affects about 10% of all global farmlands. Both these stresses reduce yield, weaken plant resilience, and also they increase the susceptibility of plants to pathogen. Drought and salinity, in fact, cause pleiotropic effects. They disturb water relation. In particular, they make difficult for plants to absorb water from a soil with a lower water potential. In this way, they alter photosynthetic electron transport and cause the increase of ROS, oxidative stress, genotoxicity, which cause membrane disorganization and decrease the capacity of plants to uptake nutrient. They alterate in this way metabolic processes and reduce cell expansion and division. All this stress together, these pleiotropic effects, cause a delay of growth and reduce yield. The effect on plants is the production in horticulture of smaller fruits with lower uniformity, visual defects, and a decrease of nutritional and nutraceutical content, with an effect on the shelf life and market quality that strongly decrease. If we go to see the effect of salinity and drought on seeds, first thing, the fact that they decrease the soil water potentials make difficult for seeds to uptake water, and so make difficult for them to have the protrusion of radicals and a first growth of embryo. because of the osmotic stress, but also the accumulation of sodium chloride and the rise of ROS cause oxidative stress and seed toxicity. And in this condition, the APA increases, thus delaying the germination. We see on the right that we can see both with drought and salinity the same effect of weakening of plants, not only on plant germination but also on growth, that decreases when the two stresses increase. The exposure of plants to these two stresses can activate stress tolerance response in against the two main secondary stresses caused by drought and salinity, which are oxidative stress and osmotic stress. Both of them activate a stress gene response and defense mechanism. This defense mechanism can be shared mechanism between the two stresses, like stomatal closure induced by the increase of ABA, osmotic adjustments, antioxidant defense that can be exerted by antioxidant metabolites or enzymes, and also membrane stabilization, or a salinic specific effect like ion exclusion with the increase of homeostasis of potassium to sodium, or the strengthening of root barriers for decrease the radial entrance of ions, but also, and in particular, the tissue tolerance, in which plant cell compartmentalize toxic ion in the vacuole while synthesizing compatible osmolytes in the cytosol, thus obtaining an osmotic adjustments. There are also some specific drought effects, like those regarding especially root architecture changes or leaf area reduction, the increase of the cuticle and wax layers on leaves, and a root to shoot shift. All these mechanisms help plants to obtain stress tolerance. However, all these defense response come at a cost. The synthesis of osmolyte and the antioxidants have a very high cost, about 50-70 moles of ATP per mole of protectant. And for this reason, when plants start protecting itself from stresses, it stops growing because the ATP, that is energy, and carbon skeletons are diverted toward protection instead of growth. Biostimulants may enhance for sure plant tolerance. It's one of the 4 effects that are mentioned in the Regulation 2019-1009 of European Parliament. And this happens because there are many formulations that of biostimulants which are designed specifically to improve tolerance to stress, and in particular drought and salinity, by activating plant stress defense. However, it is important to understand if when biostimulants help plants to increase their own tolerance, they can also avoid the decrease of growth of plants. So the good effect, the optimal effects of biostimulants would be the one of activating own plant stress defense while not reducing growth. So as you can see, there are these effects that we have in part already described for making you understanding better what is the effect, the real effect of biostimulants on plants. I will describe, I will use 4 categories of biostimulants as examples, in particular seaweed extracts, protein hydrolysates, low molecular weight molecules, and soil microorganisms. Priming with algal extracts promotes for sure the germination and tolerance to water and salt stress. In fact, this algal extracts promote the synthesis of compatible osmolytes. We see here especially for cereals, proline and glycine betaine, but proline is a common compatible osmolyte present in all plants that can rise usually after stress, upon stress in all plants, but also the increase of the antioxidant defense with the increase of the activity of enzymes like superoxide dismutase, catalase, peroxidase, ascorbate peroxidase, and their activity help protecting photosynthetic pigments, increase the photosynthetic activity, and consequently the leaf area, thus obtaining an enlargement of shoot and root and a decrease of the stress markers like ROS and MDA, that is malondialdehyde, the second one. Also in adult plants, the algal extracts can enact the same effect. In particular, also we see here the production of other antioxidant metabolites like glutathione, ascorbate, and polyphenols. And again, enzymes involved in antioxidant defense. Also in these plants, we see, in some of these plants, we see glycine betaine, but in all of them we see proline and soluble sugars. The decrease of ROS and MDA, but in previously described as the tolerance mechanism against salinity, the exclusion of toxic ions from cells or in the vacuole, and an increase of potassium to sodium ratio. In this case, when the plants can cope against stress, their capacity to uptake nutrients increases, and consequently also the nutrient use efficiency and the nitrogen use efficiency. It is interesting that there is this particular paper of Rasul and co-worker that shows that plants can exert all these mechanisms because they are able to enhance drought tolerance by also repressing the expression of a drought-induced growth inhibitor, RD26, that is an ACT transcription factor that is able to maintain cell cycle progression and meristematic cell division that usually are interrupted by stress. While if the plants continue growing, they can produce, plants can produce more carbohydrate by photosynthesis and allowing stem and root growth in a way that plants can cope better with stress. Plant priming with plant extracts or protein hydrolysate or metabolites has an important function of boosting antioxidant system and make plants able to repair DNA and progress cell division, as we have seen also before, increasing shoot vigor, germination, but also the relative water content, and decreasing ROS and MDA, and allowing germination and growth of plants. It is important to see that we see in a different manuscript in which biostimulants, different categories of biostimulants are used, that the common mechanism that biostimulants can increase are more or less the same: increase of osmolytes, increase of antioxidants. This makes plants having photosynthesis, a more performing photosynthesis, an increase of relative growth rate, but also in this case, in the case of these plants primed directly with glycine betaine, this tomato primed with glycine betaine, we have the increase of the production of heat shock protein, in particular heat shock protein 70, that is not only involved when plants are under high temperature, but also when there is a stress caused by drought and salinity, because heat shock proteins can help proteins to maintain their tertiary and quaternary structure. In this way, it is protected during the phenomenon of stress, when there is the decrease of water content in the cells, to maintain their own function. So they maintain the structure and function of proteins. So priming with plant extracts, protein hydrolysated again metabolites can promote this osmotic adjustment, can promote the control of ROS and photosynthetic recovery, but above all, protecting proteins and repairing DNA. It is easier for these plants to recover after stress, upon stress relief. Also, it is important to see here that when plants are treated with these biostimulants, there can be a decrease of ABA because there is a decrease of activity of NADH oxidase that is responsible for the production of hydrogen peroxide. And in this way, if there is this effect, there is a lower stress on plants. It's important that also single low-weight metabolites can induce an increase of plants' capacity to defend themselves. In fact, if we in this paper of La and co-workers, the priming with glutamate, the use of glutamate is able to induce the synthesis of the P5CS, that is the enzyme involved in the synthesis of the precursor of proline, while decreasing the activity of proline dehydrogenase. And in this way, we see in xylem and phloem also the increase of proline and the defense response in these tissues. Plants treated only for 28 hours with protein hydrolysate from alfalfa or animal tissues are able to increase specific enzymes against salt stress. In particular, they increase the activity of enzymes involved in nitrogen metabolism, in particular the very important key enzyme nitrate reductase, the cytosolic and chloroplastic glutamine synthetase, and in this way there is an increase of plant growth, nitrogen use efficiency, and biomass. Also, there is an acceleration of the phenylalanine lyase activity that can start the production of phenolic compounds also involved in plant stress response, and also the remodeling of root architecture, in particular the changes of lateral root number and length. So low molecular weight molecules can reduce osmotic and oxidative stress. We see here some of those metabolites that we have already spoken about, proline and glycine betaine and GABA, and but all of them are able to reduce potassium loss. So they can reduce leakage, they can reduce the ROS by ROS scavenging and causing osmotic adjustments and tolerance. All these effects are seen in an increase in the plant's capacity to increase not only growth, but also in this case, the ER sites and number of kernels, an increase of photosynthesis, and a remodeling of root architecture. If we see these main osmolyte or compatible osmolytes together, we see that they can, in certain cases, exert the same function. That is, the main function exerted by these three are that they work as compatible osmolytes. They can scavenge ROS, but they can work sometimes also as signals. But while proline is produced only in mature tissues, and their synthesis upon stress is very rapid, they are strongly linked to the nitrogen availability, but upon stress relief, they can be recycled to provide carbon skeleton and energy for the stress recovery. GABA is derived like proline from glutamate, also tied to the carbon to nitrogen balance, but its synthesis starts when there is a decrease of pH caused by stress in the cells. It is interesting that GABA can, the synthesis of GABA that happens by the carboxylation of glutamate is a process that consumes protons, and in this way, the synthesis of GABA stabilizes the pH status and pH homeostasis, but also it is important because during stress, after balancing pH in the cytosol, it can be converted to succinate for fueling the Krebs cycle and make respiration working even when there is a strong stress to produce ATP for stress recovery or adaptation. The last one I want to speak about as osmolyte is this glycine betaine, because it is produced usually only in young tissue, but only after prolonged stress, and its synthesis does not depend on nitrogen, but the strange thing of glycine betaine is that it is a metabolite that is not degraded. It can be only diluted when plants grow, so diluted to other tissues, but is a very strong antioxidant, and it has been demonstrated to protect young tissues from drought and salinity. Its synthesis is a clear adaptative accumulation, and in fact, we see here that spraying glycine betaine to plants can decrease ROS directly, ROS directly, because of its capacity to scavenge ROS, improve photosynthesis, and maintain cellular osmotic adjustments and ion homeostasis. We see here that, some microbial biostimulants are able to increase drought tolerance. In this case, we see that they are able to induce the production of daedrin, that are other proteins involved in the protection of the structure of proteins and membranes during the stress. In this way, they are able to maintain the photosynthetic efficiency, and also these microbial biostimulants can induce the production of strigolactone that are similar to ABA as signals, because they promote the growth of root and also the symbiosis with these microorganism helping plants to cope with this stress. Other microbial biostimulants can reduce the effect of salt and drought stress, but in this case, we see that they are able to decrease proline and ethylene levels. Usually, these two metabolites and gas are symptoms of stress. And it is very strange that under this strong stress, they can decrease, but these specific microbial biostimulants have a bacterial ACC deaminase that is able to degrade the precursor of ethylene, ACC, and in this way, they can reduce the symptom of stress and make plants growing better and functioning in a way that is similar to plants not affected by stress, thus producing more, thus photosynthesizing more, producing more carbohydrates, and with a very strong positive effect on plants. So what we have seen is that plant biostimulants can increase and activate the plant's natural stress response mechanism, and these plant response are for sure natural for plants, but they can enhance this response and promote in this way plant growth and functioning. And I think it is important to continue both basic and applied research on crop plants, because if we succeed in gaining a deeper understanding of the modes and mechanisms through which plants respond to abiotic stresses, we will be able to develop biostimulants that are able to better trigger endogenous response even before the stress appears. So even in absence of stress, to induce systemic tolerance in plants and help them to cope with stress without having the effect of decreased growth and reduced biomass. Thank you for your attention.
Thank you, Petronia, to you for this very interesting deep dive into plant physiology and biostimulants. But now we, let's say, shift our focus to the other pillar of today's webinar, and is biological control. So it is my pleasure to introduce Professor Luca Ruiu. Luca is a professor of general and applied entomology at the University of Sassari, and he's a highly recognized scientist in the field of biopesticides, and his work mainly deals with the entomopathogenic microorganisms and their interaction with insects. So we look forward to listen, understanding the biopesticides role in sustainable agriculture. And Luca, thank you, and the floor is yours.
Okay, thank you very much. Can you see the presentation?
Yeah, we see.
Yeah, okay. So I go ahead. So it's my pleasure to give a contribution to this webinar, and I'll be talking, as Corrado said, about biocontrol, especially microbial control, which is another piece of the puzzle. So we heard about how the mechanisms have developed, and things that are seems to be also quite complicated, and also the relationship with living organisms are very complicated. If we think, I work with pests mostly, and one of the focus of our BB-ORT project is to evaluate the capacity of microbials to act as inhibitors of the action of pests. And when I talk about pests, I especially refer to insects. If we think that insects have developed on the earth much earlier than us, and also microbials have developed a long time ago, we can imagine how the relationship may have developed in a very sophisticated manner. So I would go first to to just fix a couple of concepts regarding the ecosystem. The ecosystem is quite complex. There are different roles. There are producers like plants. There are consumers like many insects that are herbivores, and then there are microbials. Microbials often, or let's say originally, in their ancient view, are decompositor. So they have developed genes, enzymes, and way to degrade almost everything that is organic that normally we can find in the soil. And some of these microbial then have developed ways to interact with living organisms. And this is an aspect that is very important to understand how they may have developed a way to interact with insects and to overcome their immune system. Normally, each species in the environment, in the ecosystem, try to express, to maximize its biotic potential. And but of course, the population density of each species, that should be the increase of the population density, should be ultimately the main objective for any species in the ecosystem. But of course, there is an energy that is in the ecosystem that needs to be shared among different species, so that each species can increase its density in depending on the energy in the ecosystem. So there is a so-called carrying capacity that cannot never be overlapped. So there is always a fight in between species in the environment that try to find their specific ecological niches in which can develop and increase its role in the ecosystem. And now we can go to talk about horticulture, and specifically on tomato, which is a main target in our project. Tomato, there are different pests that we can consider in the ecosystem as consumers, herbivores. We call them phytophages, and they feed on the plants on different parts. Can be leaves, can be the fruits, can be the roots. What are the key species? The key species include some Lepidoptera. Especially in the Mediterranean area, we can find many noctuids, such as Heliotis armigera or Helicover parmigera, which we can find also on other crops that are close to tomato in the season. And these pests can cause big damages to the fruits. So they often go over the so-called damage threshold. So are pests that need to be controlled. Besides these species, there are also many sap-sucking small tiny insects such as aphids, thrips, alirodids that cause problem to the crop. Protection of tomato can be approached, as we have heard from now, through different methods, including the use of biostimulants, including the use of, among biostimulants, microbial. So microbial is a biostimulant, is a health promoter of the plants. So let's say that in the way microbial developed in the environment, they also learned to interact with plants to establish an equilibrium in which the plant can represent a resource, but at the same time, the plant is helped by microbials to be kept in good health. And when the plant is in good health, is more resistant to pests. But when pests come to densities which are quite high, there is no way to protect the plants just with keeping the plants healthy. This is why there is an industry of plant protection products, which is huge, which has over time developed several chemicals, especially synthetic chemicals that represent molecules that are often very effective, but at the same time, the spectrum of activity is wider, and when they are synthetic in the real world, it means that they have an ecotoxicological profile and a way to be degraded in the environment, which is much, much more difficult compared to natural compounds and compared to microbials. And microbials have developed ways to interact with insects. There are several microbials we can consider, especially bacteria, fungi, entomopathogenic nematodes, and viruses, which are not microbials, but are living forms that are able to control insects. And they are very specific. These two big groups, microbials and insects, represent on Earth the groups that show a wider biodiversity, and both of them appeared on Earth much, much earlier than us. And this diversity led to different types of interaction. There are several insects that established mutualism with microbials. Sometimes microbials help them to digest food. In some cases, some microbial developed a way to become pathogen and to go over the barriers that the insect body can offer. One of the barriers is represented by the integument. Insects have an integument made of proteins and chitins that need specific enzymes, such as proteases, chitinases, that are normally produced by fungi, like Boveria bassiana, which is one of the main species we have worked during this project. And then there are other microbials like bacteria that go through epithelia, that find ways through the gut. In a few minutes, we will see specifically one example of a mode of action, how specific it can be. It will regard one bacterium. And of course, these microbials, during the pathogenesis process, they find a response of the insect at different levels, including a cellular response of the immune system and a humoral response represented mostly by small peptides that the insect can produce. So let's say that this relationship is quite complicated. The reason why I'm talking about that is that the core of the action of microbials against insects is strictly related to the relationship they have established. So these are the group of microbials that also represents the new generation of plant protection products. So they include bacteria, such as the well-known Bacillus thuringiensis that was mentioned in an earlier presentation. But today, Bacillus thuringiensis is not anymore the only bacterial entomopathogen. There are several others that are going to be or are already registered and entered the market, including also gram-negatives. And but still, BT, Bacillus thuringiensis, remain one of the species that represent the active substance in plant protection products that has the wider share in the market. And the BT is used against Lepidoptera in tomato. So in tomato, plant protection products that are authorized for use include mostly BT. But in tomato, we find also products based on viruses, such as the Helicobacter parmigera nucleopolyhedral virus, which is a virus very specific to Helicobacter parmigera, which is this characteristic is typical and is shared among many viruses. So their action is so specific that when you have a virus active against a specific host, it is not active normally against other hosts. So this means that the market share is quite reduced on one side, but the other side it means that it's very specific, and very specific means no effect on non-target organism or on the environment. The action, of course, like similar to bacteria, is through ingestion. So there are the so-called occlusion bodies produced by viruses, and these bodies made of proteins are ingested by the insects, go into the gut, and then enter through the barriers of the gut. The viruses can enter the cell, and that's where it can be produced and can start the pathogenic process. Another group is represented by fungi. Fungi is a group in which we can find very famous species like Boveria bassiana and several Metarhizium species that represent also the active substance of different plant protection products. Their action is different, as you can see also in this picture. The fungus typically its body is made of conidia and Micelia. Conidia or spores represent the spreading phase, and this conidia is typically a hydrophobic surface, and because also the body of the insect is hydrophobic, the conidia can attach easily on the body of the insects, and when the conditions are favorable, the conidia can germinate if there is enough humidity, temperature. If the host is susceptible, conidia can germinate and produce enzymes, produce a germinating tube, an apressorium, and enter the body of the insect through the integument barrier, go inside, and that's where they can produce a second types of conidia called the blast conidia that developed inside the host. Of course, there are many molecules, and today the studies on the genome of Boveria bassiana are becoming much more important to understand the mode of action. And another group is represented by entomopathogenic nematodes. Nematodes are living organisms that we can find in the soil. There are several free living nematodes in the soil, and some of them, some other species have interaction with plants. Some is parasites of plants, but some of them are pathogens of insects. I'm using this word pathogen because this specific group of nematodes, they carry in their body, specifically in their gut, they carry bacteria. Like bacteria, normally gram-negative bacteria in the genera Photorhabdus and Xenorhabdus. And these bacteria are released inside the body of the insect in the hemocell when the nematodes enter the body of the insect through natural openings. For instance, through the stigma openings that are used by insects to breathe. And when they are inside, then the bacteria can be released. Bacteria reproduce in the hemocell that is used as a broth, nutrient broth, and they produce metabolites, and they create the conditions in the hemocell where nematodes can reproduce. Then they can leave the body, go to the soil, and go to find other hosts. So also this complex relationship that was discovered, and today we have several products in the market that can be used in horticulture against pests of tomato, for instance. I would like to spend the last minutes to say some word on the well-known mechanism of action of Bacillus thuringiensis that can just represent an example in this webinar. Bt is is a gram-positive bacterium. It produces during the sporulation phase proteins. Some of these proteins may crystallize and form the so-called crystals that contain insecticidal proteins. Today, these proteins are not anymore considered the sole virulence factors of Bacillus thuringiensis. There are several bacteria, and even the nomenclature of the insecticidal protein produced by bacteria went through a reformulation, reorganization, not anymore based on gene sequences of activity, but based on the structure, because the structure of this protein is a key factor for the mode of action. The mode of action typically take place through the gut, so the insects need to ingest the plants. Here there is a tree in this picture, but we may think of a tomato plant that is treated with a formulation that is represented by a mix of spores and crystals of the bacterium. And when they are ingested, the crystal go into the gut. And in the gut of the Lepidoptera, typically we have two conditions that help the mechanism of action to take place. One is that there is a pH that is normally alkaline, quite high, that help the proteins in their quaternary structure in the crystal to be dissolved. And then there are reducing agents that help to cut the binds between sulfur atoms. And then the protoxin that is dissolved is activated by specific enzymes like trypsin or other proteases. And then there is a very specific binding of one of the domain of the protein, normally the domain 2 for glyproteins, with receptors that are located in the membrane of the epithelial cell of the gut. As a result, these proteins, after binding, they introduce one of the domain in the hydrophobic part of the membrane. They oligomerize and create a pore. So this pore-forming mechanism of action cause a disruption of the osmotic mechanisms, and there is a flux of ions and water in the cell that will lead the cell to break. And after cell lysis, the insect, the bacterium, also the spore that is inside the gut can germinate, producing a vegetative cell, and the vegetative cells that have flagella can move and enter the insect. And with this picture that will close this representation, we can see on the left two cells. The two circles in the middle are the nucleus, in which the epithelium is well organized. And we can see also the peritrophic matrix on the left. And in the picture on the right, we can see another cell that is lysed after the action of proteins from a bacterium. So I would close this discussion reminding some main aspect that I talked about. So the availability today of microbials that are natural and are today active substances of plant protection products that are specifically authorized for use against tomato pest. And one of the aspects that we have studied, and that is a result of our project that will be in future presented, is the compatibility of these microbials with other biostimulants. So thank you very much for your attention.
Okay, thank you, Luca. Thank you for your interesting overview of biological control. And by my side, but I guess also from the audience, thank you to all the speakers, Michele and Petronia. I think that today we have covered, let's say, a full spectrum of themes in organic agriculture, from metabolic compounds, plant physiology, plant resilience, mechanisms, practical implications, biological control. And if I can give like a sort of last message, like in a nutshell, I would say that probably the future of this field lies in integrating all these technologies, from the basic science to the applied field research. So I see that we have received several questions in the Q&A and in the chat box. So I will, as agreed, Martina will manage the discussion. And bye from my side. Thank you to all of you. And Martina, over to you.
Thank you, Professor Corrado. As mentioned, we will now begin our questions and answers sessions. There are several questions raised during the presentation of Dr. Ciriello. So question number 1. Is working with biostimulant in tandem with commercial fertilizer a good starting point to grow horticulture produces on a pilot scale, a nutrient-less, free substrate?
Sorry, can you repeat again the question? I have some problem with my connection.
Sure, no problem at all. So the question number 1, is working with biostimulant in tandem with commercial fertilizer a good starting point to grow horticultural produces on a pilot scale on nutrient-less, free substrate?
Okay. Now I got it. Yes, of course. So the obviously it was not the topic of the presentation, but the possibility to apply biostimulant in combination with synthetic fertilizer. So it's something that is very very common in agriculture. So this I think that if you decide to apply microbial or non-microbial biostimulant in this specific condition, the final goal should be try to reduce the use of the chemical fertilizer in order to improve the sustainability of the entire process. And so for my experience, so this can work very well, not only in field condition, but also in hydroponics, where we usually use substrate.
Thank you very much for the answer. There are 2 more questions raised during your presentation. So question number 2, in the context of developing countries, how do you think the use of biopesticides could be viewed and by authorities and farmers who are still seeking to achieve food self-sufficiency?
Okay, this is a very tough question because as I said during my presentation, so the application or the use of biopesticides has a lot of disadvantages. One is the authorization and the regulatory aspect, but I can say that the possibility of use in that condition. So in this country, the possibility to use these biopesticides can be a useful strategy if you have a target pathogen or pest that contribute a lot to reduce the yield. And obviously, if we are able to select this pest, we can also use the right biopesticide and try to solve the problem.
Appreciate this answer as well. And there is 3rd questions raised during your presentation. If I would like to implement a project to promote the biopesticides use in sub-Saharan Africa, which kind of institutions can support me to get started? Thanks.
Okay. Probably Professor Rui, it's the right person that can ask to this question because he's more into this field. I can suggest I can answer more on the best stimulant application. That was my specific topic, but I'm pretty sure that Professor Rui can help to respond to this question.
Yes, it is my pleasure to add some to integrate this answer. Yes, there are a couple of points to keep in mind, independently of the place. For the use of plant protection products, we use this word biopesticides, but it is a plant protection product. There are rules. So in any place in the world, there are rules and there are specific authorities. In Europe, for instance, we have the health ministry in the different countries. We have the European Commission. We have the EFSA, European Food and Safety Authority. In the US, there is the Food and Drug Administration with a specific agency, which is the EPA, the Environmental Protection Agency. So each country has its own authority. So my suggestion would be first for people from sub-Saharan or other places to refer to their local authorities that for sure they have regulations. This one aspect. And normally the regulation is not only for registration